Hello everyone. Thank you for being here, and I should say thank you to the committee members for the invite to share at the symposium our little understanding about gestational choriocarcinoma. And my name is Rajendra Gharbaran. I'm an assistant professor at Bronx Community College of the City University of New York. And before I go into the discussing this cancer of the.
You're going to need to use the microphone so we can really hear you.
Before I go into the talking about the disease, I will briefly talk about my previous background. Previously, I worked on the Hox genes and neural development in an animal model called the medicinal leech. That was from my graduate work. Hox genes are genes that code for protein that regulate the identity and position of body segments along the long axis of animals with segmental body plan. I also worked on biomarker discovery for high-risk Hodgkin's lymphoma previously. Currently, I have an interest in identifying biomarkers and targets for rare and high-risk cancers. And in addition to that, I'm trying to recruit undergraduates to get, to develop a passion for this kind of work, so we can have them, starting very young. All right, and now the gestational choriocarcinoma, or GC as I call it, is a cancer that belongs to a group of neoplasms we refer to as gestational trophoblastic neoplasm, and this originates in the from the placenta. So in other words, cells of the placenta or the trophoblast can become cancerous in very rare instances. In the WHO classification of gestational trophoblastic neoplasm, or GTNs, the GC is listed as a non-molar lesion. However, GC itself arises from molar lesions such as complete moles and partial moles. And when the complete mole or partial moles develop into GC, small number of GC cases can be extremely aggressive and also highly metastatic. The frequency of GTNs, right, is much greater in South Asia, Mexico, Japan, and so on. But in Europe and North America, the frequency of the GTNs are much lower. GC itself in the West has a low frequency in the US and the West. 1 in 30,000 to 1 in 40,000 pregnancies may result in GC. In Asia, this frequency is much higher. Roughly about 1 in 2,000 pregnancy will result in GC. As we saw in an earlier slide, the GC is a carcinoma. It developed from the epithelium of the trophoblast, and they have shown that there are indication of the involvement of the syncytial trophoblast and the cytotrophoblast. I know these are some big terms and so on, but I'll explain them a little bit. In very early development in utero, this ring of cells of the blastocyst, right, around this region here, they will start to divide. The cytotrophoblast will divide, and when they divide, they produce cells that move deeper into the wall of the uterus. Those cells that move into the wall of the uterus will eventually fuse, forming this thing called the syncytial trophoblast, which anchors the blastocyst and subsequently the embryo onto the wall of the uterus. The cells that go on to become GC cells will arise from these layers here. Now, 50% of GC can develop from molar pregnancies. 25% will arise from after an abortion, 22% will occur after a normal pregnancy, and 3% can form from an ectopic pregnancy. Now it's important for us to have some kind of idea what the difference is between a normal pregnancy and molar pregnancies. A normal pregnancy, there's a diagram on the left showing the fetus present in the sac we call the uterus. The middle diagram is showing us a representation of a complete mole. In other words, there's no fetus here, and the trophoblast basically develop into something that looks like a cluster of grapes in the uterus. In a partial mole, you will have the mole present, but you also have a fetus. Now we'll talk more about how these complete mole and partial mole develop, since much of GC can form from these lesions. Oops. All right. Now, in some instances, a sperm may fertilize an empty oocyte, and resulting in a conceptus that contains only paternal chromosome. Now, this paternal chromosome, in 85% of the time, it will be homozygous, meaning it has X and X chromosome. And this phenomenon here developed into what we call a complete mole. Now, 2 to 5% of the complete mole go on to become GC. Now, the GC can also arise when complete mole giving rise to GC can also arise when two sperms fertilize an empty oocyte, resulting in a conceptus that contains only paternal chromosomes again, and in this case here, you will have the incidents that are heterozygous, 15% of these. Now, the partial moles that can produce GC will arise from what we call dispermy. That is, when two sperm fertilize an oocyte, that's good. It has the maternal chromosomes. And the resulting conceptus will contain 3 sets of chromosomes. But we have something, that product is a triploid entity. And then that go on to develop into a partial mole. Now, in the partial mole, the fetal tissue is present as well as the mole. Now, a typical karyotype of molar, from a molar pregnancy may look something like this. There are different variations of it. In this karyotype here, I think we'll see 3 copies of most of the autosomes from 1 through 22. So far what we are aware of is that the complete mole and the partial or incomplete mole can become choriocarcinoma during early gestational stages. And from here, the disease, if it's very aggressive, it can metastasize to distant organs. Some of the common areas of metastases include the lungs, the pulmonary system, and when you have pulmonary metastases, it may produce chest pain and labored breathing. You can have vaginal metastases, which can result in vaginal bleeding and discharge of, purulent discharge of nodules. You can have liver metastases of the disease also, and that can produce epigastric pain, jaundice, hepatic rupture, leading to intraperitoneal bleeding or hemorrhage. You can have, there could also be brain metastases of the disease that can produce focal neurological deficits. Now, at this point here, the question is, why wait until the disease reaches this stage? If we can stop the disease before it metastasizes, wouldn't that be good? and less worrisome for the patient, because once the disease metastasizes, and it's treated and it's so-called cured, there's always at the back of the mind of the patient that this disease can return. And one of the things we notice with some cancers nowadays, there's this thing called cancer stem cells. Well, I'm not sure if we have stem cells in GC, but cancer stem cell is a major cause of cancer and recurrence. Okay, now what we're looking at here in this display is an ultrasonograph of complete molar pregnancy. And on the right side, these are the picture of the actual moles that were formed. Now, GC, like other cancer, show there's some genetic basis that's not completely understood. First of all, we know the karyotypes sometimes are very abnormal. There have been reports of deletions and loss of segments of different chromosomes. There have been reports of amplification of the regions of chromosomes. For example, part of chromosome 7 has been amplified. There have been some a couple of germline mutations in association with GC. One of them is TP53. Another one is called NLRP7. Now, some that you may not find the same mutations in the same patient, which means that the disease might be very heterogeneous. The research on cell lines generated from GC show that a number of growth factors and oncoproteins are overexpressed. They're listed here: c-MYC, EGFR, CRB, retinoblastoma, and so on. Some of these regulate cell cycles, some of them are apoptosis regulators. And these here, if these can be validated on patient samples, perhaps we can develop drugs to target some of these oncoproteins in GC. The treatment for GC is different for the low-risk group and the high-risk group. The low-risk group can be treated with something called methotrexate and produce excellent outcome or prognosis. The high-risk group, however, have to be treated with multiple drugs, including methotrexate, cisplatin, etoposide, cyclophosphamide, and vincristine, among other drugs. The last statement there said that the multi-drug approach may produce 100% cure rate. So why work on a project like this if you're getting 100% cure rate? Well, some of these drugs here, for some of us who are familiar with them, they produce some very serious side effects that, in as much as they give the patient maybe 10 or 15 years more to live, they alter the quality of life. And can we change that? That's up to us to do the research. Now, some of the side effects of those drugs include the risk of secondary cancers, nephrotoxicity leading to kidney failure, immuno and myelosuppression, as well as neuropathy. And GC, like other cancers, may also develop drug resistance in second-line or third-line treatment after failure of first-line. Now, for some of these risks, or all of them, it's important that we generate, biomarkers and new targets for the high-risk group. Right now, the important marker that is being used for GC and all the diseases in this category, is the beta human chorionic gonadotropin hormone. It's useful in the GTNs. Its level correlates well with disease stage of GC. However, it's not specific enough to discriminate between the various types of molar lesions and the other types of cancers associated with trophoblast. And also it may not be adequate to predict whether the disease will metastasize or not. So we need biomarkers that will predict metastasis of the disease, and we need to develop targets or discover targets that will, such that if you can identify the metastatic markers, you can treat the disease very early before it metastasizes. In the hope of finding new targets, one can expect a reduction in its toxic side effects and also reduce the risk of developing secondary cancers. I think this is my last slide. Thank you. I'm sorry I finished too fast.